Guerra, and I'm a PhD student in the Hospital Ramón y Cajal in Madrid, Spain. And today I'm going to talk to you about Phytobacter, a reservoir of ESBL, carbapenemase, and metal-resistant genes in plasmids in the hospital breed environment. So, Phytobacter is a new characterized enterobacteria, formerly known as Metacosaconia, which has been described 4 species that are widely distributed and can acquire antibiotic-resistant genes as described in new recent papers. So, Phytobacter prevalence and ecology is underestimated due to limitations in the diagnostic accuracy. So in this study, we analyzed the diversity and dynamics of Phytobacter species in the hospital-built environment. This work is part of a project called Microflux, which aims to characterize the hospital microbiome and the dynamics of major opportunistic pathogens. We sampled a large ICU of 14 rooms and 4 time periods. We start in 2019 and finish at 2024. So first period was before COVID lockdown, period B after COVID lockdown, and period C we sampled the ICU empty due to the new renewed ICU, which all the patients were moved there. Then we continue the sample in period D, where the ICU was used to another medical use. We took different samples. We took 3 dry surface samples, which are the bed rails, the sink washing pot, and the touchscreen ventilator. Then we took 3 wet surface samples, which were the sink surface, sink drain, and the sink P-trap. We obtained a total of 70,887 isolates, from which 402 were Phytobacter isolates. We did a preliminary species identification with Malditov. We also did antibiotic susceptibility testing, PCR, and clonal diversity by Putzfield. And then we selected a total of 64 isolates for whole genome sequencing. We used different bioinformatician tools to analyze the core, the accessory genome, the plasmidome, and the resistome. So when we analyze the spatial and temporal distribution of Phytobacter, we see in this figure all the 14 rooms in the 4 time periods. We had 2 species, Phytobacter diazotrophicus and Phytobacter ursingi, but the Phytobacter genus can often be misidentified using Malditov. So we see in this figure that both species are widely distributed along the 14 rooms along the 4 time periods, coexisting in some places at the same time. When we see the phylogenetic relationship, we see that indeed there are 2 differential species with some clones that are persistent. Like, for example, Phytobacter ursingi has 1 clone, 2 clones that are persistent, and Phytobacter diazotrophicus has another clone that is persistent. Here is represented a heat map of antimicrobial resistant genes and a heat map of metal resistant genes. In general, what we can see is that Phytobacter has more metal resistant genes like mercury, silver, copper than antimicrobial resistant genes. Some of the antimicrobial resistant genes that we can find are in the persistent clones. Like, for example, in Phytobacter ursingi, we have BLA-TEM, BLA-BIM, and BLA-OXA2. In the case of Phytobacter diazotrophicus persistent clone, we find all of them having BLA-TEM, BLA-SHB, and some of them BLA-DHA7. So, when we look more into the plasmidome, we identify a total of 191 plasmids. When we clusterize them by relatedness, 171 were clustered in 13 plasmid groups, and only 20 were singletons, here shown. Some of the plasmids were unique to each species, but most of them were in both species. What we can see is we have a lot of IMMEF plasmids, which didn't have antimicrobial resistant genes, but they did have a lot of metal resistant genes. And in the case of this plasmid group was most isolated in the persistent clone of Phytobacter diazotrophicus, having or carrying the BLA-DHA7. Also, in Phytobacter ursingi persistent clone, we found that the BLA-BIM1 was in a persistent or epidemic plasmid called L plasmic p-oxa-48 that we did find it in other enterobacteria in our hospital sinks. So, as a conclusion, Phytobacter is a common member of the hospital water system microbiome, and it coexists with different enterobacteria that have multi-drug resistant plasmids, and this represents a risk in the persistence and transmission of antimicrobial resistant genes in the hospital-built environment. So I hope you like my poster. Thank you for watching. I want to thank all the founders and all the people that participate in this project.